Okay, folks, so for this restriction digest analysis lab, we are going to be setting up 4 different samples that will digest the lambda phage DNA with 3 different restriction enzymes, as well as a control. What we're going to demonstrate to you in this video is the setup of a single one of those restriction enzyme digests, just so you can see how it's done. Throughout this video, I'm going to try to be demonstrating excellent molecular biology technique that will make sure that we are accurate in everything we're measuring and that will prevent things like contamination. So we are going to set up sample A, or tube A, which is our HinD3 digest of lambda DNA. So the first thing I'm going to do is get a microcentrifuge tube. And again, not reaching into the bucket, I'm tapping one out onto the bench top. There we go. Putting the cover right back on, and I am going to immediately label this with an A. And I will stick it on the ice bucket for now. Oh, actually, yeah, never mind. I don't need to stick the reaction tube on the ice bucket, but everything else I am keeping on ice while we're working with it. So I'm going to be now adding my lambda DNA, my HinD3 restriction enzyme, as well as the appropriate restriction enzyme buffer to this tube. I'm going to add them in the order DNA, buffer, and then the restriction enzyme. I would say when you're setting up a reaction like this, it's good practice to always add the enzyme last, so it ends up in the perfect reaction conditions. And when I look at my protocol, I see that the volumes of everything we are adding are all under 10 microliters. So I know that I can use my 10 microliter micropipette to do all of my measuring for this. So first I'm going to add the DNA. I'm going to set the volume on the micropipette to 4 microliters. I'm going to get a tip from my pipette tip box. And careful to close the lid afterwards. I'm going to grab my sample of lambda DNA, and I am going to carefully pipette 4 microliters. So again, I'm depressing the plunger here, putting the pipette tip into the sample, gently releasing the plunger, closing the Eppendorf tube or the microcentrifuge tube when I'm done, and then adding the DNA to my reaction tube. You can see I am doing this sort of right in front of me. I'm not trying to do it at arm's length. I'm gonna have much better accuracy with my pipetting if I'm doing it sort of right in front of my face as opposed to sort of at arm's length on the bench. So I'm gonna put the pipette tip into the reaction tube. I'm gonna depress down to the first stop and then the second stop to blow out any last little bit of DNA that's in the pipette tip. And then I'm gonna press down to the third stop so I can eject the tip into the waste basket. So I've added my DNA. If I'm being really careful here, and it's never a bad idea, I'm gonna check that off on my protocol so I know that I added it already. It's always good to take notes when you're setting something up like this to make sure you know what steps you've already done. So now I'm gonna get another tip. I'm going to adjust the volume on my micropipette to 5. And I am going to get our restriction enzyme buffer. I use the same good pipetting technique that I used previously. Get 5 microliters. I'm going to add it to my reaction tube. And I'm going to eject the tip into the trash. I'm being careful to change pipette tips between every reagent to make sure that I'm not contaminating anything. finally, we're going to add our restriction enzyme. We only need to add a microliter of this. So I'm adjusting the volume on my micropipette. Going to get a new pipette tip. Making sure to close the lid after. I'm going to now take up a microliter of my restriction enzyme. So the restriction enzyme is a little bit viscous, and we're taking a tiny amount of it. So I'm going to be really careful, hopefully we can see this, to make sure that we can see that little bit of solution that got taken up. It might be hard to see in the video. Okay, so now I'm going to add this to my reaction tube, and we're good. So before we put this into incubate, I am going to make sure to spin this down in the microcentrifuge right here. And spinning this down in the quick spin microcentrifuge is going to make sure that all of the reagents are collected down in the bottom, perfectly mixed together, and we're not going to have an issue where say the enzyme and the DNA actually didn't get mixed. So I'm going to use 2 tubes, put them into the quick spin so they are balanced across the rotor, and I'm going to close the lid and just give it a 2 or 3 second pulse, and we're good. I'll take my reaction tube out, and this only has about 10 microliters of material in it, but we can see the solution down in the bottom of the microcentrifuge tube. So this is now ready to go. So I'm going to step over to our 37-degree water bath, which is the optimal reaction temperature for the HinD3 enzyme. I'm going to put the reaction tube into the tray, and we will put the lid on this water bath while the reaction incubates and while the restriction enzyme cuts through all of the Lambda DNA in the sample. In the real lab, we would have set up 4 reactions and put them all in to incubate together, but this is an example of just one. Up next, while we're waiting for this sample to incubate, we'll show you how to set up for the gel electrophoresis part of the experiment.